Okay, wonderful. Great. So thank you so much again. I'm just going to say thanks to Hilal for inviting me, and my apologies for not being able to join you all in person. So today I'm going to expand more on high resolution imaging with cryo-electron microscopy, but I'm going to fit this into context of cells and tissues. So in order to embrace complexity, as Hilal was mentioning, we also need to develop better tools to probe near native molecular structures in situ and use this as an important comparison and even a benchmark perhaps to in vitro studies. So I will cover recent challenges and advances in multi-scale imaging of neuronal systems with a focus on cryo-based approaches, which is a broad umbrella encompassing diverse research areas my new and relatively small lab works on. Oh, let's see. Okay, there we go. First, I'd like to start with a sampling of work from our lab to give you an appreciation of what can be resolved at different scales using cryo-based imaging approaches. In the top left-hand corner, this is human brain tissue from Parkinson's disease patients, imaged using cryogenic X-ray tomography. In the middle is an area from a neuron grown directly on EM grids for 3D cryo-electron microscopy. And in the corner, these are fibrillar aggregates and vesicles from a cell, also imaged using 3D cryo-electron microscopy. So now the reason why I emphasize cryo, because this is the only sibling of electron microscopy that enables imaging all the way down to the molecular level. So this is highly dependent and restricted in terms of sample thickness, but there are methods to overcome this, which I'll cover. So to understand why we cannot get to molecular level resolution in plastic bedded or room temperature on the left side, the contrast, we need to understand that contrast is imparted by a coat of heavy metals that is applied to the sample prior to imaging, and this masks the molecular structure underneath and limits the achievable resolution. So you can think of it as the process of wrapping a mummy. So you can see the shape of the nose, the skull, et cetera, which can hint at the underlying structure, but you cannot directly visualize these. So in cryo-EM, due to the nature of the sample prep, no heavy metals are needed to enable contrast, and the contrast is rather imparted by the inherent mass density of the actual molecules. So this offers a possibility to actually extracting the raw structural molecular information. So cryo has largely been dominated by imaging of recombinant proteins, but in more recent years, we've seen a move towards greater physiological relevance considering the cellular environment. We have seen beautiful papers showing angstrom level resolution of fibrils extracted from patient brain tissue material and cerebrospinal fluid material. However, cryo is not just about proteins in suspension. So although it was a big part of the original surge of papers and not the longer term, slower to evolve in situ cryo, which is actualized through a sub-branch of cryo known as cryo-electron tomography. This is enabled by correlative microscopy and cryo-focused ion beam milling, for example. So before I expand on the challenges associated with advancing progress in this area, I want to first explain a bit more about how this actually works. So we begin the workflow towards in situ cryo-EM with neurons here as an example. They're grown directly on a 3 millimeter wide EM grid. This is just a zoom in of that EM grid. So by plunge freezing these cells in liquid ethane cooled by liquid nitrogen, we can capture them in a near native frozen hydrated state. It's suspended in a glass state or vitreous ice without the use of chemical fixatives or heavy metal contrasting. Afterwards, cryoconfocal microscopy can be additionally used to localize features of interest within the cells and importantly enables us to identify the exact position in all 3 dimensions of where to create a thin lamella inside the cell using cryo-focused ion beam milling. And this thins the sample sufficiently for subsequent imaging by 3D cryo-electron microscopy or cryo-electron tomography. So since we have such a limited region of interest, a single window into the cell essentially, and not across hundreds of grid squares like with recombinant proteins that decorate the entire grid surface, we have to instead maximize the amount of information we get by tilting the sample in incremental degrees and capturing an image at each tilt step. So this way we can get more orientations of particles, which ultimately leads to higher resolution of resulting features of interest. So here on the left, you can see this is a lamella. This is a zoom in of the lamella using cryo-EM. This is completely not contrasted using metals or anything like that. You're looking at inherent mass density, I would like to remind. And F in this little panel is the coated vesicles. G is the ER associated ribosomes. And H, these are individual tubulin protofilaments forming a microtubule. So this is all in the context of their near native cellular environment. Okay, so at which steps can we improve to maximize the resolution in situ? So at the cryo-fib milling step, for example, post-milling sputter coating followed by computational removal of the sputtered platinum during reconstruction, has been found to improve resolution from 6.1 angstrom to 4.5 angstrom for large ribosomal subunit in yeast. So Kavanaugh and colleagues are aiming at sub-nanometer, like secondary structure resolution, but being able to visualize side chains and being able to fit atomic models in the sub-tomogram averages from these lamella represents a very important progress and milestone in this field, which we are testing for neuronal systems, cellular systems currently as well. So cryo-ET, I'll go back to here. So cryo-ET imaging for the second part, tilting and different tilting and dosing schemes are used, as well as combining machine learning approaches for the computational processing part, from everywhere from particle picking, machine learning and particle picking to model building. These have all contributed towards resolution improvement for in situ cryo-EM. So probing cells and tissues by cryo-EM is very limited. So we are in need of newer enabling technologies that go beyond just throwing cells or neurons onto an EM grid. So advances in recent years have been made for protein patterning of EM grids to fine-tune cell adhesion on the grids, but this is rather limited. The capabilities are limited. So to address this, my group developed a patterning system enabling compartmentalized culturing of neurons, essentially recapitulating like a very simple neural circuit towards that can then be imaged by cryo-EM for the first time. And the system is uniquely compatible with high pressure freezing for both correlative light EM and cryo-EM. So our compartmentalized clamp system for neurons, here you can see on the left, these are neurons that are transfected with mCherry or GFP. And then these EM images you can see here, these are stitched in the top right-hand corner. These are stitched together to show continuity of a single axon and higher magnification details along the axon length. So yellow arrowheads here are pointing to mitochondria. The white asterisk is pointing to axonal boutons, and the red arrowhead to synaptic vesicles, blue arrowhead microtubules. And we also have FIB data as well. For interest of time, I will show some other data. So I want to take a step backwards to the tissue again. So previously I've shown that we can dissect the ultrastructure of Lewy pathology in human Parkinson's disease brain tissue in 3D using a precise correlative approach across multiple brain samples. So here we can glean important information regarding subcellular architecture and the spatiotemporal organization of macromolecular assemblies within cells, although there are limitations that I'll cover momentarily. So we could see, this is just a zoom in using correlative TEM of one of those regions showing mitochondria that appear to be in a degradative state at the edge of this of the Lewy body shown. So we could see similar motifs in the Lewy bodies using STED microscopy, imaging markers for mitochondria in yellow, lysosomes in pink using LAMP1, and then also with phosphorylated alpha-synuclein in adjacent tissues. So we applied, why is this not going forward? Okay. Yeah, we applied a variety of complementary approaches. I just showed you the ones in blue, which likewise could be extended to complement cryofib in cells and tissues as well. So while we hinted at how these Lewy bodies may form, the reality is that we are restricted in terms of what we can say about the evolution and the maturation of Lewy pathology in situ. So to address this gap between human brain pathology and reductionist or simpler cellular systems, Hilal's team has elegantly demonstrated work that helps bridge this with neuronal models and neuron-derived fibrils, phenomena which my lab is complementarily investigating using in situ cryo-based approaches in tissues and cells. So to recap, the advantages using 2D, 3D clamp, as I've shown so far, is that we can use my protocols that are well established for human brain tissue, which is of high relevance to disease. This is insightful for helping to identify the most appropriate reductionist models, yet the gaps to fill are that this is end stage. We need to fill in what happens along the way, and we need to use cryopreps in order to achieve molecular level details about these in situ systems. So here I'll briefly show you a snippet of work that we've been doing with cryosections of tissue. These are of human brain tissue on EM grids. And this is exciting because we're visualizing mass density of completely naked ultrastructure that we can then directly apply computational approaches to resolve molecular details. So ultimately, we want to be able to resolve not just fibrils, but macromolecular assemblies in these cryo brain sections and cryo-neuronal cell systems. So in this direction, we're taking a two-pronged approach, resolving fibrils that we have extracted from cells and neurons that were originally seeded, for example, with tau from CVD brain tissue, and then imaging by 3D cryo-EM to establish a robust pipeline from start to finish for achieving high resolution imaging of these isolated components in parallel to our efforts with cryofib and cryo-ET of neurons and brain tissue. So here's just a zoom in of what I just showed you. This is on the left is cryo-electron tomography. It's a projection through the central slices of a 3D tomogram, one of 25 that we've collected. And that is more than sufficient. To put things in perspective, you usually need about 9 tilt series, and you can extract tons of data instead of having thousands of 2D images, simply because the nature of 3D tomograms, you have more information from each cube of extracted material rather than each 2D image, if that makes sense. And in the right, this inset is of tau filaments. You're probably very familiar with this paper. This is one of many. It's from Fitzpatrick. You could just look. I wanted to put this in so you could get an appreciation of similarities and the fact that we are able to see a lot of details in the cryo-electron tomogram. You can see some vesicles, and I'm just going to play this. Oh, let me play this video. I hope this works. This is just a reconstruction through showing the fibrillar structure. You can clearly see the pitch and details of the decorations and the fibrils without even having to do any serious computational processing. Now, we are doing computational processing to resolve, to get higher resolution structures, but at this stage, we can already see that there's a pitch, and we can, it's enough for us to be able to even measure the size and the spacing of the GFP densities that are decorating these fibrils, which is quite exciting. So I want to transition to this slide because not to go into too much detail about these numbers, but just to focus looking at the thickness, the emphasis on the thickness and the resolution for all these different techniques. So improve the thickness and resolution used to be the mantra for structural biologists, but now we know that thickness is not as important as knowing where to target, for example, which new technologies as cryoconfocal microscopy can enable, for example, and using technologies such as what we've developed for cryocompartmentalization of distinct neuronal populations, which helps to better capture complexity for in situ cryo-EM. Lastly, I also want to emphasize that higher resolution in vitro has stood alone for a long time using cryo-EM, but we are now at the stage that we have technological capabilities. We're really at the wavefront here to add in situ context using 3D cryo-electron tomography. And lastly, that cryo-ET can provide important information without having to go to sub-angstrom resolution about the subcellular milieu in a near-native and frozen hydrated state. So lastly, I'd like to extend my thanks to collaborators, both past and present, and generous funding support, most currently from Parkinson's and Parkinson's Foundation, NIH, and most of all the patients that were involved in the Lewy body part of my talk. And thank you very much again for your time.
Thank you, Sarah, for a beautiful talk. We have time for questions. David?
Yeah, thanks for that lovely talk. A couple of technical questions. What sort of substrate do you use to grow the neurons? And do you need a cryoprotectant in the freezing process?
No, we do not need a cryo. That's an excellent question. Sorry if I cut you off. Did you want to say something more?
I wondered what sort of substrate you're growing them on.
So we are growing them directly on holy carbon. So there's no cryoprotectant used. There's no additional substrate used. We are also in parallel growing them on graphene. So the problem is once you add more substrates, substrates in terms of for the neurons itself, PDL is what we use for the neurons. And I believe Matrigel for different cells, we just use the thin coating, but we don't use anything beyond that like Form-Var or things that would interfere.
And how about glow discharge?
Yes, that's a great point. They absolutely do need to be glow discharged. We have tested different types of glow discharging settings because glow discharging, there's a fine line. If you glow discharge too much, you're going to blow the holy carbon off the grids. If you glow discharge too little, the grids will be too hydrophobic and no substrate will attach to them. So we optimized glow discharging parameters that allow the neurons to adhere very nicely to the EM grids.
Thanks so much.
Yes.
Any other questions? Okay, I have a question, Sarah. I was wondering if you had followed up on the Lewy body work in terms of looking at other pathologies and whether you see the same level of complexity in terms of composition of pathologies, vis-a-vis the presence of organelles with other pathological aggregates in different neurodegenerative diseases, or is that unique to Lewy bodies?
That's an excellent question. Actually, I have not investigated further. I believe Amanda is doing a lot of that now in Henning's group. She's looking at she's exactly looking at that. So I cannot personally say it. I've moved more of my work focusing on the cryo and really trying to establish the workflow very nicely for cryo in situ work. But that's an excellent point.
Okay. Thanks again for looking forward to following your work. So.